Assalamualaikum and hello everyone. I hope all of you are doing well despite the busy schedule that we're in right now. Okay, so I will be explaining to you on how we can conduct practical 5, which is multiple sequence alignment. In this practical, we'll be able to know how to do a multiple sequence alignment using several servers or software. Okay, but before we start, I just want to let you know that there are some objectives that we cannot achieve because there are some server or software that is no longer available or have already retired. Okay, so yeah, let's start. So first of all, let's dive into the background information. So what is multiple sequence alignment? A multiple sequence alignment is a sequence alignment of three or more biological sequences, generally protein, DNA, or RNA. In many cases, the input set of query sequences are assumed to have an evolutionary relationship by which they share a linkage and are descended from a common ancestor. Okay, so why do we do multiple sequence alignment? Usually, MSA is a tool used to study closely related genes or protein in order to find the evolutionary relationship between genes and to identify shared patterns among functionally or structurally related genes. So, what's the difference between multiple sequence alignment and pairwise sequence alignment? Is that in pairwise sequence alignment, and it uses an alignment method which compares two biological sequences of either protein, DNA, or RNA. However, in MSA, it uses an alignment procedure comparing three or more biological sequences of either protein, DNA, or RNA. Okay, so let's start with practical 5.1, which is multiple sequence alignment using biology workbench interface. This is what I meant by not having the server anymore. The biology workbench, if you search it on Google, you will see that it's no longer available. So for this practical, we'll only be doing from step 1 until step 8. Okay? So the point of practical 5.1 is that you want to find out what kind of gene it is for this given accession number. Okay, so when you find out the gene, you will need to perform a signal P to locate the signal peptide. Signal peptide is a signal P is wait I actually drew a picture for you. Sorry if it's a bit messy. So for example, let's assume that this is the cell, and the circle right here is the nucleus. Okay, and outside here is the proteins which are produced outside. Okay, so signal peptide sequence usually will be located at the either C terminal or the N terminal of the protein. Okay, C terminal or N. Sorry for the messy drawing. Okay, so for example, there is an mRNA, and there is a protein coding sequence. This is the protein coding sequence, and the red one here is the signal peptide sequence. So what we need to do is that we want to remove this signal peptide sequence. We need to remove this. Okay. Signal peptide is a few residues of amino acid. They are responsible in sending signals to the cell telling them where to translocate the protein. And so signal P can tell us that location of the signal peptide sequence, this location in the mRNA, so that we can remove it. Okay, so this is because we only need the protein coding sequence, which is this one, to be converted for the multiple sequence alignment. if signal peptide sequence is included in the MSE, it will reduce the accuracy because it is a highly variable region. Therefore, in order to generate MSE, protein of different identity is used by removing the signal peptide. Okay, so I'll close this now. So after you remove the signal peptide, you will achieve the mature lipase. Okay, so this mature lipase, you will need to paste it on BLAST. After you click search at BLAST P, it will give you a few sequences. You know what? I'll just show you how to do it first so that you'll be able to understand it more. Okay, so first of all, you search the accession number AY260764. Okay, so go to the NCBI and search for AY260764. So you click search. And it will give you this the result, and the result shows that this accession number represents the thermostable lipase gene. So what you need to do is you want to click FASTA to copy the whole DNA sequence of the lipase gene. So we need to copy all this, click copy. And then, based on the practical here, it says that you need to perform a signal P. However, when you open signal P, when you open signal P, you'll notice that they only accept protein sequences. And the one that we got earlier is in a DNA form, is a DNA sequence. So we need to translate that DNA sequence first into a protein sequence. So we need to go to XPC and paste the DNA sequence that we copied earlier and click translate. Okay, and then when you scroll down, you will see there are many frames. Okay, there are about 6 frames, and 3 frames represents the 5' to 3' direction, and another 3 frame will represent the 3' to 5' direction. However, in this practical, we'll only focus on the 5' to 3' direction because we assume that the transcription occurs from the 5' to 3' direction. Okay, so these are all the DNA sequence that has already been translated to amino acid residue or protein sequence. Okay, and you can see there's a red highlight. Okay, this red highlight represents the open reading frames. So when you look closely, you'll see some similarity. You'll see a similarity, which is that all of these open reading frames start with the letter M. Okay, this M represents the methionine. Okay, methionine is also known as a start codon. So this open reading frame will always start with the start codon and ends at a stop codon. So among this open reading frame, we need to choose or copy the longest open reading frame and paste it at signal P. Why we choose the longest is because the functional protein is usually the longest polypeptide because it needs to fold properly. So when you look at this frame, it's very obvious that the second open reading frame is the longest. So we need to copy that and then paste it at the signal P server. Okay. So you paste it. So this is already in protein sequence. So you click submit. Then signal P will give you a result in a form of a graph. Okay, this is the graph. Okay, so in this graph, as you see, it provides three lines which present three scores. Okay, the first graph, the first line, which is the red line here, is the S score, which gives us the end of the signal peptide. S score is highest at all positions of amino acid residues until it reaches the end of the signal peptide or the cleavage site. Okay, so from here it starts to decline, and here at A is where the signal peptide ends. Okay. So next is the C score or the Y score, which is the yellow line. Okay. The Y score gives us the range between the beginning of the signal peptide and the end of the protein. So as you can see from here, it starts to increase from here until here. So we assume that this is the end of the signal peptide, which is around A here. So this is the range of the signal peptide, sorry. And lastly is the C score. The C score gives us the first amino acid of the signal peptide. So how we're going to read this is that by looking at the highest peak of the C score. Okay, so the highest peak represents the first amino acid for the signal peptide. Okay, so if you combine all of these three scores, we'll be able to estimate the signal peptide sequence. Okay, so after looking at all the scores, we know that the signal peptide sequence is located here before A until the end, until the first amino acid residue. So what we need to do is that we need to remove that signal peptide to achieve the mature lipase. So you need to count manually how many amino acid residues starting from E. So I've counted it and it's around 27 amino acids. So we need to go back to XPC, the one that we translated, and we need to recopy the gene. Okay, and since I've already calculated and the gene started from the one that I highlighted here, which is A at the 28th amino acid. So here into here. So we will copy this. So this is what you call the mature lipase gene. And this mature lipase gene will be pasted on this protein blast. Okay, so you need to go to blast and click protein blast. And paste the mature lipase gene that we retrieved just now. Click blast. Okay, once you click blast, you'll get a list of sequences producing significant alignments. Okay, so we need to select only 10 sequences with varied identity. Okay, based on the result, we can see that the lowest percentage identity is around 93%. Therefore, we need to select at least 10 to 12 sequences. So the 10 sequences will be selected within this range from 93% to 100%. Though our report says that we need to select 2 from each percentage, but since the result only have 93% percentage identity and above, we can only select the 10 sequence from that range. Okay, so you need to select the gene manually. So what I look at when I want to select the gene is that I'll make sure that I won't select the sequence with the same percentage identity, since we want a varied identity. Okay, actually I've already selected the gene, and if for example if you have selected your genes like 10 genes, or 10 sequences, you can click here, download, and you need to download it in FASTA format. And since I've already done it, this is my FASTA format. Okay, so you can see there's like 10 sequences here, Geobacillus, blah blah blah. So this is the 10 sequences that I've selected. We need to keep these sequences so that we can use it for in the multiples in our next practical, which is practical 5.2 until 5.4, where we need to use like different servers for multiple sequence alignment. Okay, so this all these multiple sequences has not been aligned yet, so now we have to align the sequences, try and align the sequences. So first, the next practical, which is practical 5.2, we are going to use Clustal W. So when you click the link Clustal W, you'll see that Clustal W is have services have been retired. So instead of using Clustal W, we are going to use Clustal Omega, which is quite similar to Clustal W. The only difference is that Clustal Omega is a combination of progressive and iterative method. So you click that. Okay. So you click that, and you need to paste all the 10 sequences that you selected from Protein Blast earlier here to do the multiple sequence alignment. So from here, I'm going to copy all of this. Sorry. I'll click copy and then I'll paste it at Clustal W. So, and then I click submit. Clustal W, since it is a progressive method, Clustal Omega, since it is a progressive method, the result will show it is quite a fast process. So the result usually will show immediately after you click submit. These are all the multiple sequence alignments. So if you want to see it in an easy way, you can click show colors, and so that you can read it easier. So this asterisk here represents the conserved region. So at the region where it has asterisk, it means that region is conserved. However, and you can see this part here, it doesn't have asterisk. This is because it is not fully conserved, because there are sequences that is not similar to the rest of the sequence. Not all of the sequence have similar regions. So, and then you can click on results viewers, and then click on gel view to colorize your output. Actually, in order to use gel view, you need to install it in your desktop first, and I've already installed it, so I'll show you what it looks like. So this is the result that we get from gel view. So the function of gel view is only to colorize your output. So you can click on color. You can choose what is it that you want to focus on. For example, hydrophobicity. Okay, it will show you like a range of hydrophobicity. Okay. Conservation will show you the quality, consensus, occupancy, all this, cluster X. So yeah, that's it for the Clustal W. So now we'll move to the next practical, which is Praline. Okay. So Praline, if you read this practical, it needs you to display the result in different formats, which is the rescue type, conservation, secondary structure, and hydrophobicity. So these four formats are actually the characters or properties of the Praline. So if you get these, it tells you the application apply consistency approaches. Okay. It is hard to do the same analysis using progressive because progressive puts the sequences bit by bit, so it will lose consistency. But if you do a consistency-based approach, which is Praline, it now looks at the design of pattern. So if this protein share a particular residue, it will align the residue. It doesn't do step by step. So I'll show you by opening Praline right now. So this is Praline. So similar like the one we did in cluster W, just paste the gene that we have obtained from practical 5.1. Just copy the whole gene. Okay. Copy that and then we paste it in here. So and then we click run. So the thing about Praline is that it takes a long time for the result to show up. So compared to all the server in Practical 5.3, which is Cluster W, Praline, and Espresso, Praline is considered the slowest server, while Cluster W or Cluster Omega is considered the fastest. So you can actually bookmark the result and bookmark the page and come back later to see the result. But since I've done it earlier, I've already bookmarked it. So this is the result. So when the result shows up, you can immediately see the alignment score, like percent sequence identity, number of sequence, alignment length, number of residues, and number of gaps. So when you further scroll down, you will see results color coded for amino acid. Okay, so you can choose the characters or properties here. This one here is the conservation. This is the residue. For the residue, okay, this is for the hydrophobicity. Okay, and as you can see, the color it is labels the hydrophobicity of the amino acid. So from the baby blue until the red ranges from hydrophobic until hydrophilic. So you can see which part of the sequences which have similar hydrophobicity. Like for example, you can see here. Yes. So and next you can open when you display results in different format, you can see the 10 to 12 sequences secondary structure, which I just open now, that you share the sequence or not. So for example, okay, it says here that there is 3-state secondary structure for each sequence is represented by a color. If a sequence in the alignment has no color assigned, this means that either there is no DSSP information available or that no prediction was possible for that sequence. So what DSSP here means is that it is a secondary structure prediction method to locate the secondary structure. So from here, the red color code represents the alpha helix and the blue one represents the beta sheet. So you can see, based on the color, you can tell if the secondary structure of your sequence share that sequence or not. Okay, if they have the same color, it means that they share the same sequence. Okay, submission. Okay, so this is conserved. If you look here, the red means it is fully conserved, and as you can see from all of our 10 sequences that most of the sequences are fully conserved. Only at the beginning part is slightly different. Okay, and also about the secondary structure earlier, if all your protein shares secondary structure. This secondary structure means that it is annotated to a function. That function can be a pocket for your lipid or catalytic site in which the enzyme breaks down the lipid. Okay, so this is the lipase chain. So that is it for praline. And finally is the espresso. So espresso incorporates all the available structural information in your alignment. Okay, so what it means here is that espresso is a structure-based approach. Okay, earlier praline, I told you that it is a consistency-based approach. Okay, praline is a consistency, while espresso is a structure-based approach. Okay, espresso is progressive. It means that it's faster than praline. However, there's a problem about once a gap, always a gap. So the sequence at the end usually have a lot of gaps. However, in praline, uh, since it uses consistency approach, it has the same amount of gaps. And you'll also be able to look at the residual, conserved site, secondary structure, which is, the character outputs and such. So let's open espresso. So when you open the link, you need to click espresso, and same like the rest of the server, you need to copy the sequence that we got from practical 5.1. So espresso is quite fast. However, it is not as fast as cluster W. Like you've seen earlier, cluster W will show the result like almost immediately. However, in espresso, you still need to wait like a few minutes in order for it to show the results. And I've also bookmarked the result, and this is the result that I've gotten. So what espresso does is that it takes all your 10 to 12 sequences that you choose, and it will do multiple sequence alignments based on structural info. Okay, for example, if there are 2 to 3 sequences with no structural info, this server will exclude that sequence from the multiple sequence alignment. So what you need to do if that happens is you need to go back to BLAST and select another 2 to 3 sequence that was excluded to replace it. Okay, so in order for you to know if all of the 10 sequences that you've selected share the same structure or not, you need to click this one, template list file. Okay, so so all of this sequence shows that, for example, if I count, there's 10 sequences, and the sequences that I've selected were at 5.1 was also 10, which means that all of my 10 sequences is structurally aligned. Okay, so yeah, I guess that's it for this practical. I hope you understand. If there is any question, feel free to ask me. And yeah, okay, thank you all, and goodbye.